Hello everyone. I made this video to provide you with some feedback for the second rough draft so that you can incorporate this into your third rough draft and be on the pathway to be writing an excellent paper in May. So this is just general impressions from the whole class, and just to make sure that all of the requirements are explicitly explained so you know exactly what to do. So a lot of people have started writing the results section. In the results section, you present the data as a narrative that describes the logic of the experiments as a sequence of figures or tables. The figures have to be accompanied by a paragraph. So you'll have a narrative with subsections. Again, the JBC format is sans serif, no initial caps. So the paragraphs in these subsections should constitute a narrative in which you tell a story. So you shouldn't be repeating experimental detail here, because if the reader wants to know that, they can go to the experimental procedures and find it there. So in your narrative, you're just telling a story, and you're calling out the figures and tables, as seen in figure 1, or as shown in table 1. So just write a story, and as you write a story, refer to your figures and tables as you write your story. For example, results, again, so look at this, bold, sans serif. So the subsections are bold, italics, sans serif, and no initial caps. So this is a screenshot from a recent JBC paper. So this is how they began their results section. So just to provide an example of what it should look like. The figures should be on the figures on top and the captions in the bottom. And notice it says figure 1 with a period, and the caption is bold sans serif. They have an explanation of the figure. Our explanations don't have to be that long. You can just provide a brief explanation and make a shorter caption, and then the explanations can be in the text itself. But there shouldn't be any kind of data analysis in the figure caption. This is another screenshot of a JBC paper. So what you can see if you look on the top here is you can see where I screenshot it, that the text is in columns, but once the author gets to the figure it goes all the way across the page, and the figure caption does too. So you shouldn't be squashing your figures into columns. It doesn't help the reader, and it just distracts from your narrative. So you need to have your columns, you need to turn on and turn off the column function as you're doing this. When you present your data, you present only your data. So I told you before, I was like, I don't want to see this picture or anything like this in any of the papers. The other thing you do is you're going to renumber the lanes. So lane 1 is the standards, and then whatever lane you happen to begin in becomes lane number 2, whether it was lane number 2 here or not. It doesn't make sense to do it any other way. Label some of the bands on the ladder, which I've done here. Which I haven't done here, which you should do, is our plasma is 6292. So you should have another arrow right here that points to 6292. For your results section, if you plan to present anything in tables, and when you get to the kinetic data like this, you will. The caption is above. So figure sits below. Table sits above, so just say table 1, it gets its own line, bold sans serif, and then this is the next line, sans serif, not bold, not italicized. That goes on its next line. So if you do tables, that's the format for the tables. At the end, we have our experimental procedures. So right now, what's happening is people are starting to write introductions, results, and experimental procedures. So I saw some papers that had the experimental procedures at the beginning. So you're not going to present it to me in the order in which you did it. It's going to be the order in which it's going to finally appear in your paper. So it appears at the end of the paper, lots of detail. If the reader wants to duplicate what you did. And a lot of times when you read a paper, you're not really interested in that. So that's why it's at the end. But at the end, this section should still tell a story. You should have subsections, and they should naturally flow from one to the other to tell your story, explain what you did in a logical order, concisely and succinctly. So it has to be brief, but it still has to be complete. So that's a fine line to walk. Remember, your audience is biochemists who have experience working in the lab but are unfamiliar with this particular project. So do not tell them how to do certain things. So here's another sort of snippet from a JBC paper. So again, this is how they began their experimental procedures. So experimental procedures, bold, sans serif, no initial caps, and then they're in subsections. So here we have bold, italics, sans serif, no initial caps. And then they start their narrative right here for this subsection. So that's what it should look like. So you should be doing the JBC format here. If you wait to the end, you will have a lot to do in May. You don't want to do that. So start doing it now. So the subsections need to be in this order. So in terms of what we've done, in terms of who has contributed to your paper, there are multiple contributors. So I am one. For some of you, students at University of San Diego is another. But this is what makes logical sense to talk about our project. You start with the bacterial strains, and then we prepare liquid cultures. I prepared wild type liquid cultures, but I'm an author, so that needs to be there. You did mutant liquid cultures. So whatever you describe in this subsection has to be specific, but it has to be generic enough where it applies to both the wild type and the mutant. And we typically do it the same way, so that shouldn't be a problem. Same thing with the plasma mini prep. Because you or I did a plasma mini prep for the wild type, and then you did the plasma mini prep for the mutant. But again, when you describe it, it should be something that applies to both. So basically what we're doing is you have bacteria, you prepare a liquid culture, you prepare a mini prep, and now you have this wild type plasmid, and what you want to do is you want to do a mutation. But first you'll do their multiple sequence alignment, so you should describe that. And then since that's informed the mutant that you want to make, then you're going to describe the site-directed mutagenesis. You will do a plasma mini prep, but you're not going to explain it twice. So all you have to do is just say mutant plasmids were mini prepped as described above. That's all you have to say. And then you are going to send it off for sequencing. And we will add to this list later because some of you have already done this. We'll do a transformation of the XJB cells and do the protein overexpression. So this list is going to get very much longer in the coming weeks, but at the minimum, this is what we have to have for now. Starting with bacterial strains. This is from a non-JBC journal, so I showed this in class. But it does have this model of somebody who was given a gift of something that's not commercially available. So our cells are E. coli. It's a strain BL21DE3, and what I got in the mail from Dr. Bell was an LB plus can plate streaked out with these cells. So they were cells with a plasmid, and the plasmid expresses the human MDH1 variant 3. So that's what we got. So once I get something like this, then I can start a liquid culture and then do a plasmid mini prep, and then I can purify the plasmid, the wild type plasmid. Liquid cultures. So again, this is a non-JBC journal, so it doesn't have the format. And they do some things that I don't agree with. Don't capitalize the names of the antibiotics. Do specify all of the ingredients in the LB broth because there's sort of different flavors of LB broth. Don't use the phrase routine concentrations, please. Okay. When you prepare the liquid cultures, again, write it sort of generally to include the DH5 alpha cells containing the wild type and the mutant plasmids. List all of the ingredients in the media. Don't provide a recipe. Just give the final concentrations. Don't use the phrase LB plus can. We use that in our notebooks. It's a little too informal for a paper. And again, we talked about this in class where only successful preparations are described. The exception here is that there are 3 people who started with plasmids that were prepared by students from last year, so who tried to do double mutants, and none of those were successful, unfortunately. What we should do there is you should describe your attempt to make the mutant plasmid, and then when you get to the point where the sequencing showed that the double mutation was unsuccessful, then you simply say the decision was made to carry on the investigation with a single mutant, and then you just go on and describe what you're going to do. But anyway, liquid cultures, E. coli, capitalize it. Capitalize the genus, don't capitalize the species, italicize the whole thing. And don't spell out Escherichia coli, just say E. coli. Everybody knows what it is. Again, the plasma mini prep, write a nice narrative. You need to cite the name of the kit, the catalog number, and the name of the company, but it doesn't have to be this long, protracted thing. All you just have to do is say, Simon Research, Irvine, California, Zippy plasma mini prep kit, and then whatever the catalog number it is. And because this is a narrative that has to make sense, you should say initially that you followed the manufacturer's instructions, and then you should list the modifications. So some people just went right to the modification, and you're reading it, it kind of doesn't make sense. So you have to start with, most of the time, we did actually follow the manufacturer's instructions. And there's a link here that you can find because this PowerPoints posted on Canvas, you can go to that link. Consult Simon's protocol, see what you did differently. And again, just report the successful sample that you went on with. This only applies to the people who did the restriction enzyme. That's only 2 of you. And again, report the number of units and report the total mass of DNA in micrograms, the digestion temperature, and the time, and that you did electrophoresis. Again, just report the percentage agarose, and you would report the time that you ran the gel, the voltage, and the CyberSafe stain that we used to visualize the DNA. Okay, so when you choose your mutation again, so now we have our plasmid, we have it's a wild type, and now we're going to write how do we choose your mutation. We're going to do a multiple sequence alignment. So no detailed descriptions, no analyses. If you have any of that in your experimental procedures, you can move that. That will go in either in your results or discussion. So your time doing that hasn't been wasted. You just have to move it. You will reference Dr. Provost. He was the first author on this paper from Essays in Biochemistry, because here, what's novel in this paper, it's mostly a review of what other people have done. But what's novel in here is that one of the co-authors in this paper, because I was on this paper, but I didn't do this, but one of my co-authors did a really deep dive into the various databases that predict phosphorylatable serine-3 and tyrosine. And because that was the most recent, other people have done this, but that was the most recent analysis, you should cite this paper. And down here is, again, this is from a non-JBC journal, but this explains how someone would go about describing what you did. Okay. Site-directed immunogenesis, the narrative is good. There's stuff in here that I don't want you to do. You should spell complementary properly. Don't refer to tables or figures in your in experimental procedures. Don't use volumes. Don't use 10x. I want final concentrations only. So just report the total mass of DNA in nanograms and the concentrations in micromolar of your primers. And again, we followed the kid pretty precisely. There aren't that many deviations. You do need to include the primer sequences. This person uses future tense. Please don't do that. The whole paper should be past tense. You're carrying out this investigation, and then after it's over, you're describing what you did. So everything's got to be past tense. So nice narrative from primer design to sequencing. Describe your logic. Reference New England Biolabs with the catalog number. The primers are from IDT. You do have to report the sequences either here. You can have a table in the results sections. I prefer to do it that way. But do not. I told you this the first time, and people are still doing it. I don't want to know how you prepare your stock solutions. IDT provides this information. The reader can get it. So don't mention this at all. The reader can get it from IDT. The other thing I saw is that people talked about ordering equipment or supplies. The word order should not be in your paper. Do a search for it and get rid of it. Okay, for back to site-directed immunogenesis for the PCR, sequence of the primers, final concentrations only, no recipes. The entire thermocycler program should be reported, but not the hold. The hold is just for our convenience if we just happened to not be there when it was over. Usually we were, and we'd stop it, and then we would just go on with the experiment. But you just cool things down to preserve them. So delete that step. Again, with the KLD, we pretty much followed everything. So you do need to describe sort of the logic of what you did, but then you have to describe with, well, after that, then we did we added the SOC and the cells, and then we proceeded with plating after that. So again, logical narrative so someone can follow what you did. More examples from the JBC, so I will let you do that on your own. Again, look for a search for X. I don't want to see 1X, 10X, 6X. I don't want to see it. Delete it. More examples, and again, here there's an example of how someone would report a PCR program. And again, notice that they didn't have the hold, so delete the hold. And again, here's the protocol. You can find this on New England Biolabs. And but again, they have a very generic, this is routine PCR. You can't report that. It's got to be exactly what you did. The only exceptions here are the people who are doing the University of San Diego. They actually have to just say that routine PCR was used, because I don't actually know what they did. whatever they did fit into these parameters, but I don't know exactly what they did. I have to ask my colleague for that information. So if you checked your results by running a gel, that's not part of the NEB protocol. We didn't do this there's at least one person that doesn't have that information because I did the site-directed mutagenesis. I actually didn't check because I just didn't have time. If you choose to report this, that's fine. You don't have to. But again, if you do, you would want to include the gel. Don't include a recipe, just as 1%. TBE buffer, so again, all the components. Don't explain how you prepare your samples because you would just use the 6-hexyloidine dye, and that's we got from NEB, and that would be included in the manufacturer's directions. And again, crop the gel. It shows your results only. So again, and then renumber the lanes so that it makes sense. It doesn't make sense to have lane 1 and then lane 7. That doesn't make sense. Okay, so now what you did is you did a plasmid mini prep, but since you already described it for the wild type, you can just say that the plasmid mini prep for the mutant plasmid was prepared as described above. So you do need to then say that you sent this out for sequencing. Again, just include one sample, indicate the sequencing primer used, and then just reference GENEWIZ. Okay, don't do this. No recipes, no descriptions of routine procedures. I'm still seeing this, and we talked about this last time. People are saying, like, explaining to the reader how to inoculate a culture. Assume they know how to do that. People are showing us like how to operate a NanoDrop. It's not a machine. NanoDrop begins with N. And they might have something else, so it doesn't make sense for you to include the operation of our NanoDrop since theirs is probably different. It's most likely different because our NanoDrop is probably 20 years old. They probably have a nice new one. And no information on storage samples. I want that for the notebooks. I don't want that in the paper, unless it's something critical or something not well known. So for example, once we get our purified protein, you can't freeze the enzyme where it dies. And that's not something that's commonly known. So that's something like that you'd include. But something like freezing DNA, well, that's something everybody would do. So don't include that. No links, no recipes, no analyses, no storage condition, no dishes, no tubes. So just do a search for tube and just get rid of it. No plates, no pipettes, no Bunsen burners, no inoculating loops. Just anything like that doesn't belong in experimental procedures. So we're putting references in, and we have references within the experimental procedure section in terms of we use a kit. So you need the name of the kit and the catalog number, the name of the company, and the location. Those references should not appear at the end of your paper. The other thing you can do is once you've referenced a company, you can be brief the second time, like for Zymo, for the plasmid mini prep, you could reference Zymo completely, but then we got the XJB cells from Zymo too. So you could just say Zymo Research and be done with it. You already gave the reader the information about the company's location. So again, for the kits, start out by saying that the manufacturer's instructions were followed and then list the deviations. That's a narrative that makes sense to the reader. Company references, IDT. So all you have to do is you would say when you refer to your primers, then just say obtained from Integrated DNA Technologies, Coralville, Iowa, done. Just in parentheses. GENEWIZ, Azenta Corporation, done. So reagent cells, reference the company in parentheses. That's all you have to do, and those references should not appear. I did a search on Zotero to make sure that those references weren't there, just to make sure I did not tempt you. But please don't have them appear at the end of the paper. That's not where they go. Now we are putting references at the end of the paper now, and we know how to use Zotero to do that. So Pymol example is in there, and the Provost Essays in Biochemistry is in there. So I also put a reference in there to the Leninger textbook because for the introduction, we're starting out by describing MDH and just sort of reminding the reader of the biochemistry that they might already know, or maybe with biochemistry, maybe they don't know. But I put that on Zotero too. So I have you on as viewers and not as editors. So if there's a reference on Zotero, you can actually grab it yourself and put it in your paper. That's something you can do easily. If you don't know how to do that, I can show you. But if there's something that you want on Zotero, I can publish it. So tell me, just give me the DOI and I'll put it on Zotero, and that way everyone in the class has access to it. Your document, there's a word editor. You can check spelling, grammar, all of that. Genus and species names are italicized. Antibiotics are uppercase. Concentration used must be correct. I think people are not doing this. I don't want to see a refrigerator or a freezer. So again, you report the temperatures and not the thing that is providing that temperature environment. I see a lot of create. Get rid of the use of the word create. Maybe your theology professors want that. I don't. We are not using the word create. We don't create anything in this class. Don't start a sentence with a number. That's just not something you do. Get rid of that. I've seen still a lot of things that are informal, like protocol followed. It's the protocol. I mean, you can have something informal when you're talking to your friends or you're in your notebook. That's fine, but this is a formal paper. It needs to be formal. Past tense at all times. Spaces between numbers. This is too hard. If you do 5 mils, like this is just too hard to read. So put a space between the number of the unit. And again, no X's. Do a search for X. Get rid of it. You want to make sure that you're not copying from protocols, websites, published protocols, things like that. That's going to be particularly important when we get to our introduction. So I want to see prose that sound like you and not like somebody else. So like at the end. So I still see a lot of people are trying to use, I don't know, fancy verbs, especially. There's some verbs that are they're just weird. It's like, just use a verb that's just normal and common and plain and simple. Don't try and be fancy, because usually when you do use a verb that doesn't make any sense and really doesn't convey the meaning that you want to convey. So don't do that. Read your paper out loud to yourself and see if your narrative makes sense, or give it to a friend and see if they think it makes sense. So start using your JBC protocol now, because if you wait before the paper is due, then there's just one more thing you have to do, and just do it now, and it'll just make your life easier in the end. So I hope this was helpful. If you have any questions, let me know, and see you in lab, and thank you for your attention.